for joining us for day 3 of CFDE Pediatric Power Up. All right, so thanks so much for joining. So first of all, I want to introduce a new face joining us today. So this is Rowan Beck. She works at Vilsera. She is a great partner of ours who helps build a lot of cloud-based platforms like Cavatica and Biodata Catalyst. And so she is available to help us in case our demo go off the rails today. We think we're going to keep it on track, but just in case things turn sideways, she's available to help us out today and tomorrow. And then she's also available. She's going to come in on Friday as well, so we can talk about any questions about using the platforms or where to go next, or basically how do you take what you learned from this course and whatever kind of level you're at, how do you take it to the next level. So thanks so much for joining today, Rowan.
Yeah, thanks for having me.
Yeah, of course. So to recap and say where we are and to look forward, we're on day 3 of our workflow here. And so we started with a very kind of basic introduction to what is the Common Fund data ecosystem. And so it was this introductory level of what is CFDE, what are these different organizing centers, what are these data sets, and how do we get access to the data. So it's very kind of all about getting started and learning about the types of data there are. So once we learned about the data on Tuesday, Sangeeta gave us a great presentation about, well, what are all the different things that you can do with it? So what are the types of analyses that you can do, and how do you analyze and look at this stuff? And so today, continue to kind of build and scale and get larger and larger. We're going to do some analysis on some data from Kids First, and we're going to look at ways to combine data from GTEx with it as well. So today, we're going to use an existing app called DE-seq2, which is going to allow us to do some analysis on bulk tissue RNA-seq data that's available through Kids First. And then so we're using a tool that's already made. We're going to set it up and click some buttons, and then we're going to look at the output at the end of it. On Thursday, we're going to go one step further. This is tomorrow. So Sangeeta is going to be presenting again, and she's going to go the next level is, well, how do we build a pipeline? So not just use an existing one, but how do we set one of these up ourselves? And so she's going to be looking at some single cell RNA data that's available through the HubMap program. And then, as I said, on Friday, we're going to wrap it up. And so we're going to talk about, well, how do we go next? Now that we've introduced these platforms, maybe you have your own research question. How do I, move your project or your needs forward? So that's what we're going to go on Friday. So that's where we've come from and where we're going, and we're here right in the middle. So we're going to start looking at actual analysis. So our agenda for today is, I've got a couple slides at the beginning to briefly introduce some concepts about RNA-seq, and then we're going to log on to the Kids First portal to find some files of interest. Then we're going to run the DE-seq2 analysis tool in Cavatica, and then we're going to talk about how 2 different ways that you could incorporate GTEx expression data with that. All right. So some very basic intro. So I know Sangeeta mentioned that there are different sequencing technologies, and I don't want to restate all the things that Sangeeta presented, but I do want to highlight, as we're talking about RNA-seq, some of the concepts behind this analysis that we're talking about. So without talking about all the chemistry behind high throughput sequencing, high throughput sequencing data produces small little individual pieces of DNA called reads. And so we don't, unless you're doing long read technology, most sequencing data comes as these very short 75 to 150 base pair segments of the genome. And so, of course, the human genome is much larger than 150 base pairs. And so each of these individual puzzle pieces need to be put back together in order to be able to assemble the genome. So this is what some of this high throughput sequencing data looks like. These are called reads. And so here's these are 76 base pairs long. So we've got, here's the letters that represent that 75 base pairs in the genome. And then it's got some quality score information And then, so these 4 lines correspond to 1 read. And then these 4 lines correspond to the next one, and so on and so forth. So these are what's called unaligned reads, or this is in what's called FAFQ format, which is kind of the most basic, uninformative, unuseful state of the data, because right now it's just kind of, you can imagine the analogy I like to do is it's a bit like puzzle pieces that are still in the box. So together they will, put together this puzzle of a whole genome, but right now we don't know which puzzle pieces go together. We don't know how the whole genome kind of assembles. So that's what kind of the preliminary work that's done is taking these reads and puzzle piecing them together into a genomic alignment. And so that works a bit by finding, overlapping pieces, just like you might do on a puzzle where, these have a similar color or a similar pattern. So I'm going to put these puzzle pieces together, or try to fit these puzzle pieces together. That's what the alignment computer software is doing as well. It's looking at these reads and finding sequences of DNA that look similar, and then, puzzle piecing them together. So for example, if this read ends TATGGC, that lines up with this one that has TATGGC, and then it's able to, you can assume that, oh, those 2 pieces go together, and now we have a slightly larger piece. And then we can continue to kind of scale and get bigger and bigger and bigger. And so these are alignments. And so we get this information about each of where these puzzle pieces go. And we're starting to get more useful information. You can imagine now we have a completed puzzle instead of just like pieces in the box. And so today and tomorrow, we're going to talk about RNA sequencing. And so Sangita also mentioned several times the central dogma. So to go back to high school biology class, we've got DNA, and then it's transcribed into RNA, and then it's translated into protein. And so each of these molecules can be captured, and you can collect information out of them. And so today we're looking specifically at RNA and RNA sequencing. And so how that works is that you'll collect, a researcher will collect their RNA, their tissue, extract RNA out of it. Since RNA is an unstable molecule, they'll actually reverse transcribe it back into DNA and then run that through the sequencer. But you can imagine, based on the central dogma, some genes are more highly expressed than others. And so if a gene is highly expressed in a particular tissue, then it's going to have lots of copies of RNA floating around inside the cells, and then we would have lots of protein that are being produced at the ribosomes as well. So what the RNA sequencing is doing is it's quantifying that RNA molecule. It's saying how many of these molecules are there in the cell, and it's using that quantification in order to estimate how highly expressed particular genes are. So kind of a rough schematic of how the concept works is that we have a collection of samples. So here we have 2 samples, the blue sample and the purple sample, and they're both loaded onto the sequencer, and then we align them against the genome. So because these are RNA, they're only going to map to genes, because they're, the puzzle pieces are only going to fit together where we have those particular genes. And so you can see for sample 1, we have lots of reads here on top of that map to gene A, and a couple of reads that map to gene B. Whereas we compare that to sample 2, and there's less. So there's fewer of these reads mapping to gene A and to gene B. And so we can infer, if we are properly set up the experiment and run the appropriate amount of RNA through the machine, and are controlling based on thousands of genes in the genome, we can infer that in sample 1, gene A has a higher expression. So it has 30 of these reads, whereas gene A in sample 2 only has 10 of these reads. So it's 3 times higher. There's 3 times more level of expression in that particular sample. So what that gene is, so here we're just gene A and gene B, but that could be any number of things. That could be a transcription factor that's activating expression of other genes downstream. It could be some type of oncogene that is driving the cell cycle that might be proliferating a cancer. And so we would want to then look into, well, what is gene A doing, and what if it is so highly expressed in sample 1, and does that correspond with any hypotheses that we might have about our particular experiment. So this is what we're going to be looking at. So today we're going to be looking at some of these expression fold changes as we dive a little bit deeper into the data set. All right, so that was my brief introduction to the kind of the methods and the science and the logic behind RNA-seq. And so we're going to look at some RNA-seq expression files that are available on the Kids First portal. We're going to push them to Cavatica, which is the cloud analysis platform. We're going to set up the DE-seq2 app. We're going to view some output from the app, and we're going to talk about ways that we can bring GTEx data into Cavatica as well. All right, so I'm going to stop sharing my screen now, because now I'm going to switch over to my browser. And I will show this window right here. All right, so let me share the screen again and share this window. All right. So today I'm going to be largely following the directions in this tutorial right here, which let me pull up. So many windows floating around. Here we go. Here's the chat. Largely going to be following this tutorial, which shows how to do RNA-seq in Cavatica. So there's my directions if you want to follow along later, and we can send that as part of the follow-up for the course. And so the first thing we can do is we can log on to the Kids First portal. So we can go to kidsfirstdrc.org. And so when we go to kidsfirstdrc.org, sorry, I'm just making sure all my windows are in the right spot. When we go to kidsfirstdrc.org, we can log into the portal where we're going to access that particular data. So let's see, maybe I can log out so that way I can show. So when you navigate to this page, it will ask you, it gives you some overview information about what's on the Kids First portal, what different studies, and then we can click login. It gives you a couple of different options to log in. I have kind of the recommended method is that if you have an NIH ERA Commons ID, then you should log in with that. If you don't, that's okay. Anyone who has a Google authenticated email address, like any Gmail address, can log in and create an account as well. So I have my ERA, so I'm gonna click this button, and it's letting me know that we're gonna access it through that. I'm gonna allow them to do it. And now we are live on the Kids First portal. So I am going to kind of connect some of my studies here real quick, just to make sure. So I've already been approved for access for a couple studies. And so I'm going to confirm that. And I'm going to do my NCI studies as well. And we're going to allow that. So that's just the authorization. And we're going to close. And then I'm going to refresh my screen. And it should pull up. So now you can see these are a couple different studies that I have data access to. So the kind of the overview of the method that we're showing here is that, so the Kids First portal is really based around kind of like a search and discovery type of tool. So it's very good at looking things up. It's good at finding files. It's good at searching for things. And it's good at kind of, that kind of discovery step of, well, what is out there and what types of data exist and that sort of thing. And so the portal is paired with Cavatica. So the thing about the portal is that you can't do too much analysis on it. So you can kind of just look and find the files. But if you want to like touch the files and explore the files and open them up, then you really need to use Cavatica for that. And so the portal and Cavatica connect very well and very easily, as I'm going to show you here today. So that allows for this kind of transfer from one space to another. So at this point, I think I'm actually going to jump into Cavatica and we'll get started over there. And we can explore a little bit further. So I'm going to go to our Cavatica URL, which is cavatica.sbgenomics.com, and I can drop this in the chat as well. And once again, it gives you two different options. I have my NRH ERA, so I'm going to log in with that. But again, anyone can create a username and password with any email address, so you don't need an ERA comments to create an account. All right. And so when it loads up, we're going to click here to reload the page. Perfect. And so when it loads up, you get a couple different options here as far as it shows some projects that you've been working on, as well as some analysis, different recent tasks. So mine probably looks a lot busier, especially if this is the first time that you're logging into the platform. So yours might be blank for each of these two squares. But Cavatica is based around this concept of projects, and each project has its own apps, its own data, its own collaborators. And so everything is kind of nicely organized into that little project space. But then you can have many, many different projects for many different collaborators, and they all kind of combine together. So we're going to create a new project right now. And we're going to call this Power Up Demo. You can call your project anything you want. You're provided with some pilot funds to help support you in the beginning. I'm going to use this CFDE based training project billing group, and I'm going to create my project based on this. Create. So I mentioned the billing group, so maybe I should discuss that a little bit further. So what we are doing here is, so what we're trying to do is we're trying to do these very large computational tasks. And so you can imagine, like I showed with the my puzzle piece example, it's easy when there's 5 DNA sequences, then you can kind of line up the letters and very easily kind of map them together. But what we're asking these computers to do is we're asking them to take thousands, perhaps millions of reads, and assemble this puzzle back together for us, which is a very computationally intensive task. And so you need a very powerful computer to be able to do that. So that's more than most traditional laptops or desktop computers are capable of doing. And so what Cavatica does is it makes available much more powerful computers up in the cloud that we can basically log on, borrow the computer for a little bit. The computer will run the task, and then it will return the output to us. And so as we're borrowing the computer, it costs, there's a small cost associated with that. And so it costs some money, depending on how long the machine is running for, and we're billed based on that. We can talk a little bit more later in the workshop about estimating costs, good ways to kind of keep your costs down. And then also we've been promoting the cloud credits program through CFDE. And so there's an opportunity for you to use the platform, and then NIH will flip the bill to pay for your work. So opportunities there exist as well. All right, so that all said, so here I am in my new project with my username. There I am, smiling, so happy to begin my project with some description here. So you can actually edit this. You can change this little box. You can write some notes here to yourself. You can say hello world, and now we have that. So you could use that as like a place to jot notes or a description of what we're doing in this project. So right now, our project is pretty empty because it's brand new. So we don't have any files in here. And this is, as it says here, files are the basis of every analysis. So we can't do any RNA analysis until we get some files. So there's a couple ways you can add files. You can add them, upload them if you have things on your computer. You can kind of drag and drop them through an upload, which we'll actually show a little bit later. You can pull them from a couple different repositories using GA4GHDIRS, which we're going to show a little bit later with when we're looking at GText. You can also connect volumes as well. So if you have your own, cloud infrastructure, your own AWS or Google storage, you can connect it as well. And so I'll also highlight while we're here, this very helpful button down here at the bottom right. So this is the blue question mark. And so this is a really awesome question mark because when you click on it, it gives you links to the Cavatica documentation. And so it gives you information about, oh, I see you're on the files page. And so it gives you a couple different links about, well, how do I, maybe you need help uploading files, or maybe you need help tagging your files. And so it gives you a couple different links, and then it gives you these great detailed directions about how to actually do that request. So if you're ever stuck, the question mark, or the helpful question mark, is always there in the bottom. You can click it. If you get further stuck, you can go to the homepage of the Help Center, and they will give you some, help you find the directions. Or you can also type in this box. And hopefully you can, share more useful explanation about what you're trying to do and where to go. And then if you click the send button, it will go to Rowan and her team, so that way they can help you kind of resolve your issue. So just wanted to highlight that feature. It's a really useful and great one about Chromatica. All right. So we need some files. So let's get some files. So we're going to get our files. So I shared that there's lots of different ways you can do it, but we're going to get ours from the Kids First portal today. So I'm going to go back a tab, and we're going to go onto the Kids First portal. So we can review our different data sets by going to the Studies tab. And so as we mentioned, there's lots of different Kids First studies. Right now we have 35 on the portal. And all of them have genomic data, and then a handful of them have transcriptome or RNA sequencing data as well. So today for our demo, I'm a little limited by what I can show on like a public, sharing my screen demonstration. So I don't want to show any controlled access genomic data. I'll get in trouble for doing that. So what we're going to show open access transcriptome data, where we're just looking at counts of gene expression that anyone can access. And so that's why I've chosen to do RNA sequencing as our demo here today. So you can see that if we click on our studies tab and we click on just the studies that have transcriptomics, then it narrows down with these 6 studies are the ones that are possible candidates. So today we are going to look specifically at the CVTN brain tumor study. And so we're going to do a comparison where we're going to look at 2 different brain tumor types, and we're going to compare gene expression. And so, you can imagine that, the 2 tumor types that we're going to look at are the medulloblastoma, which arises in the cerebellum, and ependymoma, which arises in the epindal cells, also in the central nervous system. So these are 2 different brain tumors. We would definitely expect them to have different gene expression. We're kind of going in with an expectation that things will be different here, but we're going to choose this as our example today of what we're going to be looking at. So we're going to be looking at the Children's Brain Tumor Network as our project here today. And so I'm going to jump over now. So we're going to explore our data. And you can see, since I was in here last night practicing, that we've already got this query, but I'm going to delete this query and we can recreate it once again. And so there's a couple different ways that we can search for data on the Kids First portal and narrow it down. So we already know that we want to look at this particular study, this Kids First Children's Brain Tumor Network study. And so we can do that by, if we just want to look at that particular study name, we can add a filter for that. And we can type in Children's Brain Tumor Network, here it is, and apply our filter. And so the way the Kids First portal works is it kind of works like a library, like digital catalog. So if you're looking for, an article or a book, the library probably has thousands of books. And so you would add in, well, it's by this author, and it was published in this year, and this is the title. And so you can kind of narrow down the thousands of books and just try to find the one book that you're looking for. So here we're doing the same thing. We have thousands of files on here, and we're going to kind of narrow it down to about 100 or so files that are relevant to what we want to do. All right, so now we're looking at CVTN. So if we scroll down, we have some information about what types of diagnoses are represented here. So we see a lot of different pediatric brain tumors, as well as some medulloblastoma samples and some ependymoma samples. And so that's good, because that's what our demo is on today. We can scroll down. We also have some information in a different ontology. This is the human phenotype ontology, which also is sharing us similar numbers, that we have a broad collection of different tumor types in these participant samples that we're looking at today. All right, so then when we get to the bottom, we have a couple different file types, and so let's try to figure out what files we need. So today we can see that we have some we have participants. So this is the number of participants on the x-axis here. So we have some participants with genomic DNA as well as participants with transcriptomic DNA, but it's not an exact one-to-one. So we are interested in the RNA, the transcriptomics. So we just want to filter based on only if you have expression data. If you just have a genome, then today we're not necessarily interested in those particular files. So we can add a filter a couple different ways. We could go into data file, data category, and click on transcriptomics. But the other way to do it is actually you can click this bar right here, and then it will add the filter as well. So there are 881 participants with transcriptomic data in the Children's Brain Tumor Network. So now what we want to do is, well, we're only interested in, you can see by looking at our diagnoses, we have lots of different diagnoses here. And so we want to just hone in on two. We want to hone in on medulloblastoma and ependymoma as part of our demo. So we're going to select those by looking at a biospecimen filter. And we're going to add a histological diagnosis mondo. And so histological diagnosis, what this means is this is a specific histological condition that's been assigned by a pathologist who reviewed a sample and has assessed that, yes, this particular cancer tissue is the medulloblastoma, or it is a DIPG, or it is a low-grade glioma. So we are going to select medullos because that's part of our demo. And then we're also going to look at, let's see here, ependymoma. There it is right there. Apply. So now we can close this. And so now you can see I'm getting progressively longer and longer with my query here. So we're looking at CBTN transcriptomics, where the sample is either ependymomas or medullos. And we've got 115 participants, so that's pretty good. And we have 1,110 files. So looking through here, we can see that, kind of scrolling through. So 1,000 is pretty good, and this might be the number that you want. But we're going to try to pare this down a little bit further, because if we scroll through here, we can see that there's actually a couple different types of files. So we see that there is BAM files, which are those alignment files that I talked about. So that's the assembled, puzzle pieces. And so we can, if you're interested in doing that RNA seq analysis where you're running through, you're counting, how many reads are at these particular genes, then you would want to start, with the alignment yourself. And so likewise, we have some other files in here. So this is a PDF file, which hopefully everybody's probably familiar with. And so a PDF of different gene fusions. So RNA is also used as kind of the other use for transcriptomics. So not just looking at gene expression, but also looking at which transcripts might have fused together due to a chromosomal break. These are especially helpful in cancer samples because cancer, one of the hallmarks of cancer is these large chromosomal breaks and rearrangements. And we've also got so we've got some of these fusions, we've got some alignments, and we have, in fact, I can maybe add, let's see here, if I add a column for data type, it will tell us a little bit more about what these are. So we've got some fusions, some alignments. And so what we're interested in is this type of file right here, this gene expression quantification. So what this file is we've already done that analysis of counting, how many reads at gene A, how many reads at gene B, how many reads at gene C. And we've already done that per sample, since that's what many people, want to start to do. So we're interested in these. We're not going to start today from the alignments. Instead, we're going to start with the gene expression files, and we're going to go from here. So we're going to add one more filter for this gene expression quantifications, and that's going to pare down our files even further. And that should be our last filter we're going to add, gene expression quantifications. Da, 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 da. All right, so we've got 244 files. So that feels pretty good. We're going to continue to filter a little bit more in Cavatica, and I'm going to show you how to do that. But first, we're going to move these over there. All right, so I'm going to select all of these files. So if you wanted to be, just choose specific files. And actually, you can tell by looking at them. So our file name here, we've got this code, and then you can see some of them end in callisto.abundance, and some of them end in rsem.genes. And so what these different words are, Callisto and rsem, these are two different bioinformatic tools that we use as part of our RNA pipeline. And so these, as I'm moving my window here, and so each of these tools, each of this data comes from our own RNA tool. So we do this bioinformatic workflow, which we make available on GitHub, and I'll show you. We also keep a copy in Cavatica as well. So this is where these files were produced from. And so these tools, rsem and Callisto, are two of the outputs, or two software tools that make outputs that we ultimately make available. And so, what this tool does is what exactly I was describing. It reads in this kind of intro of the alignment file, and then it runs it through this software tool called STAR, which does the alignment, and then it has these tools, these two different tools, rsem and Callisto, which counts how many reads are at each of these different genes. And it also has a couple of fusion callers as well. So this is our tool, and this is where all these files came from. So if you're wondering, well, what is the context of these things I'm seeing? This is kind of a connection here. Okay, so we actually only want the Callisto files as part of our tools here. So we're going to select all of our results, all 244 of our files, and we're going to click Analyze in Cavatica. So now we're going to copy them. So we are allowed. So first it's going to check and make sure, do you have access to this? Because like I said, so a lot of the data on the Kids First portal is controlled access. But we specifically chose to do this RNA expression project because these are open access files. And so anyone should be authorized to copy them to Cavatica. So I am going to click the Analyze in Cavatica button one more time. And I'm going to click, where did my new demo go? Let's see here. Maybe if I refresh my screen. And so it's going to save my query there. So you can refresh, and we're not going to lose all that work we did. So let's select all again. And we're going to Analyze in Cavatica. And there we are. There's our Power Up demo. So we're going to click Power Up Demo, and we're going to copy the files. And so while it's doing its work, and in fact it's already done, the word there that we use on the portal is copy. You are authorized to copy these files, and then you click the button that says copy the files. And we use that word copy because that's kind of the analogy that folks understand, is that, oh, we've made a copy and now it's available. If we click over here. So here's my Power Up demo project. If we said, oh, there's no files here, but if I click refresh, then ta-da, here are all those 244 files that we just copied over. So a word about the word copy. So we're not truly copying anything here. So there still exists, wherever this file lives up in the cloud on an AWS server in Northern Virginia. The file, it only exists as one particular file. But what we've done is we've copied kind of a little pointer. You can imagine like a shortcut that you might have on your computer that points to that one copy of the file on the cloud server. And so so we're not truly copying, we're not proliferating, more and more files and creating more and more storage. We're using the power of the cloud to keep, our number of, instances and copies of these files all to its one particular spot. So here are my files, and you can see we've got that mix of Callisto and RSEM mixed in here. All right. So now we've got files, so that's great. So now we've got to do some analysis on it. So we can do that by looking at the apps. So I'm going to go over here to apps. And right now I don't have any apps in this particular project. So just like files, the apps are unique to your particular project. And so if you have multiple files, then you need unique apps as well. So how do we get our apps? Well, we can create an app, and we're going to explore that a little bit further on tomorrow when we learn about building this single cell tool. But today we're going to keep it a little more basic, and we're just going to use existing tools that already exist on the platform. So to do that, we're going to add apps from public apps that already exist. So this is just like the app store that you might have on your phone. You can scroll through and find the different apps. But the best part is that these are all, free to access. You don't need to, sign up to use any of them. So I'll show a couple different before we hone in on the one that we're going to use today. I'll show, for example, so first, so I mentioned that our tooling exists in Cavatica as well. So all of the files on the Kids First portal, to go back over here, our new query, all of our 28,000 participants and 173,000 files, we made all of them on Cavatica. And so we use the tool a lot. As I click to the right page, there we go. We use the tool a lot, and we, kind of believe in it as well. And so in order to do that, we make available, well, this is how, not just we did it, but this is how we did it too. And so these are our bioinformatic tools. We put them up in the public apps. Anyone can use them as well in order to be able to do their own analysis. So here is our Kids First workflows. So we've got our alignment, some structural germline SV workflow that we're starting to put together. We've got our long reads workflows, which we built last year. And so we're excited to get those going as we have new long read data. And then if we go to let's see here, RNA-seq workflow right in here in the middle. So this is the same tool as this GitHub repository. And so what we've done is we've built it here in Cavatica with the same commands that allows you to be able to run this same tool just like we did when we made these files here on the Kids First portal. And so the useful tool of this is that it allows you, if you have your own data, where you have your own RNA-seq data from your own research project and you want to combine it with the Kids First data to have a bigger analysis, you can do that. And likewise with the CFDE, if you're want to combine CFDE data sets, but they're not perfectly harmonized just yet, which is a big thing that we're working on in CFDE. But if they're not harmonized just quite yet, if they're not run with the same pipeline, you can take that CFDE data and run it through the Kids First and get it all to be harmonized, and then you can combine it together for analysis. So this is here. We're not going to run this one today. We're actually going to use the output of this this workflow. So this workflow, if we read it, gives us some introduction. Tells us this is that same. You can see this is the same kind of description here, but now we've produced it here on Cavatica, and it tells us about the particular outputs. If I scroll down, it has some directions. So if you want to run it yourself, it gives you some kind of tips for this is how you do it. And then this is all the different types of outputs that it produces. So we're actually going to be using some of these files, this Callisto abundance file, which is one of the output files that it produces. All right, so we've already got our abundance. So you can imagine we've already run this. So this is helpful because we don't need to run this big workflow again. That's going to save us time and it's going to save us money, which everybody in scientific research appreciates, I'm sure. So we're going to back out. So we don't need to run this today. So we're going to go back to the App Store. And so we can go into, if we add our apps and go into our, let's see here, how should we do this? I guess we can go, let's take a little break here. And we're going to go into the Data Studio. No, let's continue with the apps. So we're going to pull in the project that we want from the public app. and we're going to pull in DESEK2. So DESEK2 is a this kind of workhorse of a tool that allows for differential gene expression analysis across different study conditions. In fact, we can click on it and learn more about. So it gives you information. So what this DESEK2 actually is, it's actually kind of an R package. So normally you would be feeding these files into an R, our studio environment, and then you would run this R package to be able to produce this output. So what this app is, it's in Cavatica and it's on all Vocera platforms, is that it actually allows you to be able to kind of run that DESEK2 without having to spin up the your RStudio environment. So we are going to look at the output in R a little bit later. And so we feed in a couple, it gives you some information about the science behind the workflow. And then, really similar to the kids' first ones, the authors of this tool, they give you some information about what you input, what is the output, what does the annotation files look like, things like that, so on and so forth. So this is our app that we're going to set up today. So we're going to click the Run button. And so when you click the Run button, it's going to ask you to copy it to a particular project. And we're in our Power Up demo project. So we're going to click Run. And so now, if you've been following perhaps carefully, we've actually been kind of moving, progressively further along this little bar up at the top. So we started with the dashboard, which kind of introduces us. And then we looked at our files and we've collected our abundance files here. And then we looked at apps. And now we have copied this DESEK2 app to our project. And then now we're going to run it. And so every time you run the app, that's what Cavatica calls a task. And so this is one instance of running the DESEK2 app. So we're going to click on this. And so it asks for a couple different inputs and then some settings. And then after it runs, it has some outputs over there on the right-hand side. So the first thing it's asking for is expression data. And so it even gives you some helpful hints here as far as what that particular thing means. In fact, it recommends for transcript level abundance, use the results from one of these tools. It's bioinformatic tools, so they all have fun names, so salmon or sailfish, but we have Callisto here today. So we're going to choose our output from Callisto. So to do that, I'm actually going to go back to our files. So in our project, we have a mix. So we actually have multiple files from each sample. So even though we have, I can't see my count here because my windows are in the way, 244 items here, we actually have 2 files per sample. And if I pull up our particular, let's see here, I think perhaps case ID, nope, information, then it would tell us that we have, because we have one RSEM and we have one Callisto. In fact, you can tell because this is the code that we have this .rsem and this .callisto. So this represents the same kind of analysis, it's just run with 2 different tools. So today we're going to run with the Callisto. So we're not going to use the RSEM. So we actually just want these particular files. So we can do that. You can use the search tab up here. And we can type in KILL, or I guess we can do it a couple different ways. We can search for Callisto and then enter. Searching, thinking about it. And what it will do is that it will basically searching all the 244 files in this project, and then it's going to just select those, and then it's going to hide all the RSEM files. Once it thinks about it a little longer, and I might refresh my screen to give it another try. Let's try again, let's refresh. It's thinking very carefully. And take a drink of water. Here we go. So now you can see we've got, so we had 244, but now we have 122 selected. So like I said, we have half. So that meets our expectation. And all of these are just our Callisto files. So what we're going to do is we can do it a couple different ways. So we can select all of the files in our project, and we can add a little tag, just a label, so that way we can find these files more easily. So I'm going to add a tag, and I'm going to call these Callisto add. And so now what it's doing is it's adding a tag for these 122 files. And so basically this is kind of like a bookmark in your browser. And so if you want to be like, show me all the things that have this tag, you can apply this tag very easily, and it will pull that up for you. So if I close my search, so now you can see I've got this little tag by every other file for Callisto. That's great. And so I can search based on my tag, and I can pull up those files much more rapidly. So that's good. I'll also add in some additional columns here. So for our columns, so we can add additional kind of metadata. And so part of some of our updates, I think we have to check about how the metadata is being loaded in Cavaticas, which is something I need to follow up on today. But I'm going to add in the histological diagnosis Mondo. So you'll remember that was our search term that we were searching for earlier. And so if I scroll over, so here's that search term. So we have a good mix of medulloblastoma samples as well as some ependymoma samples mixed in here. So there's our, kind of our two kind of comparison states that we're going to look at. So we've got our files. We could even go back, so I can search for, so if we search for files that have no tag, so this is kind of the opposite. So this is all of our RSEM files. So we could keep these in the project. They're not like hurting anything. They're not taking up any storage space. But I'll also show you that you could actually select all these and we can delete them out of the project. And then they're just not going to bother us anymore or get in our way. So try to keep things tidy. So then if I hit refresh, and of course, if I had screwed up, there we go. So there's all, now we just have 122 files and we just have a Callisto one. So that's great. If I had ever screwed up and if I had deleted stuff that I really needed, then you can always go back to the portal, select them again and push them back again. And so we can just, kind of make that new copy or that new shortcut whenever we need it. Okay. So now what we're going to do is we're going to go back to our task and back to our DE-seq2. And we are going to select, so we need to feed in 3 different files here. So this is the expression data file. So we're going to select files. And so we're going to select from all possible extensions. And so here's all our Callisto files. And so we're going to select all of them and we're going to feed them into the workflow, save the selection. And so now you can see loaded up here, we've got all of our kind of input files. This is these counts, 1 per gene, and we've got them loaded up, ready to go. So then the next input that we need, is gene annotation. So what is this? Gene annotation tells us the names of genes based on gene IDs in our input. And it will help us summarize it for gene level analysis. So that sounds good. We want one of those. In fact, we need one of those, and it won't let us go forward until we add in this file. So this is a file that's in this .gtf format, which is a pretty standard format. And so we're going to add in our gene annotation file that's associated with our particular, how this RNA data was called. So we can go back to that RNA workflow. And we can look for, the input for our tool here is this .gtf. And so we could download the file and re-upload it, but instead we've already got copies of these GTF files that have already been loaded. So I'm going to go to select our file. And so we don't have any GTFs in our current project. Remember, we've just copied the Cavatica for the Callisto output from the portal. So we're going to go over to projects, and we're going to go over to public files. And so public files, I think Sangeeta introduced this yesterday, but public files is this really useful space where a lot of the references are kept. and such that, you don't have to like find it on the internet or search or try to figure out which one you're looking for. So we are specifically looking for a GTF. We have 11 different files, and so we are going to add the suggested one for today, version 27, which these particular files will run on. So it says that we're going to add this to our Power Up demo project because we need to copy it to our space to run it, and that sounds good. We'll allow that. Ta-da! So we're 2 out of 3. Okay, so now we've got our annotations loaded, we've got our expression data loaded, and so now the last tool we need to add, the last piece we need to add is our phenotype data. So this is a CSV file with phenotype data for all samples. So each sample gets its own row, and then metadata in subsequent rows. It's crucial that these sample IDs match those in the metadata of the expression data files. Okay. So what we're going to do is we are going to create that file by going over to our tab here. And so, so we need to create this file for the phenotypes. And so we're going to select all of our list of files and say, see, now we have that extra annotation file. So we're going to drop that one. So we have 122. And so now we have our abundance expression files. And we are going to go to the ... dots over here, and we're going to generate a metadata manifest in comma separated. So what this is doing is it's basically taking this big table that we have here. If I move my windows around, I can scroll over. So it's taking all this information in this table, and it's just saving it as like a comma separated file that we can download and then could open in like Microsoft Excel, for example. So if I refresh my window, ta-da, there's our manifest that we just generated. So we're actually going to download this to our computer now. I'm going to show how to do it in Microsoft Excel because that is what our guide that we're following today has. So you download the file, but you see I'm only downloading one small file. I'm not downloading hundreds of very, very large files. So we're doing pretty good as far as keeping our data in one spot. All right, so I'm going to open up this file on my computer, and so I've got to exit the browser real quick. And then I'm going to open a tool called Microsoft Excel, which you're probably familiar with. And we're going to open this file that I just downloaded. So let me open this up, and then I'm going to share my screen again. Microsoft Excel. And I'm going to share this window. Okay. So this is the manifest that we just downloaded. And so it has some information about the nature of these particular files. It's got this ID column. It's got the name of the file, some size. It's got information. So there's our histological diagnosis Mondo. So there's our selection that we were choosing. So some are medulloblastoma and some are ependymoma. And it actually has some additional information here as well about the nature and the biology of the tumor. So many of these are initial tumors, whereas some of these are progressions of the same tumor or they're recurrences of the tumor, at a later time. So for our tool here, so it says that we need sample IDs in the very first column there. And so we are going to use this, let's see here, external sample ID. So we're going to use this as our sample ID. So we're going to copy this column, and we're going to go back to the beginning. And we're going to paste that column. And then we're going to call this column sample ID. And so that gives us information about the particular sample name that's associated with this particular files. So now we're going to save this. And so that's all we need to do. So I'm going to save that. And then actually, let's go back. Let's add in, instead of deleting that column, let's add in a new column. So let's see here. Now we're learning how to use Microsoft Excel on the fly. We're going to insert a column. Perfect. Insert cells. And we're going to call this sample ID. Perfect. All right, we're going to save this. So we've saved it, and I'm going to give it a new name just so that way I know what it's called. So we're going to save it as, we're going to call this, because it's the phenotypes file, we're going to call it phenotypes, and we'll call it power up. And we're going to save it as a comma separated value, save. All right, so now we've got this file saved, so we're going to stop sharing, and we're going to go back to the cloud and back to Chromatica. So switch back to the browser, and which is this window right here. All right, so now what we're going to do is we are going to go back. So now we need to add that file up into here. So we're going to add our files from my computer. And it says you can just drag and drop it, or you can browse. So I will browse for it. And there's it is, phenotypes power up, open that. And so from here, we can then start the upload. It's a very small file. So it should take, it's already done by the time I'm done with the sentence. So we're going to close this. And then the upload is finished, and it says to refresh. And ta-da, there's phenotypes power up. Okay, so this is our file, our last file that we need. I'm going to try to do one more thing here. So if I select these three and I go to metadata, update values, let me check one thing. There's a way. All right, update values. All right. I'm not going to try to do this right now, but so we should have all the pieces we need. We can now take this file and we can go back to our task, and we can go to DESeq2, and we can add the phenotype data file for phenotypes power up and save the selection. All right, so now we are good to go. We have all of our inputs. We can go with some settings now. So we can give this a title. We can call this power up. We can say, so what is your covariate of interest? So this is the critical one. So this is which of these different groups are you trying to divide? You know, you want to compare apples to oranges. What? So or boys to girls, or old, you know, participants to younger participants. And so this is where you want to like draw that line. So for our particular demo here, remember we're drawing the line as histological diagnosis Mondo. So histological diagnosis Mondo. So that's that field that has either ependymoma or medulloblastoma. We're going to update the FDR cutoff value. This is the p-value that it declares significance. So we're just going to update it to 0.05. And then our quantification tool, this is asking what software tool did you use to make the files. And there's our handy friend Callisto. So we're going to click Callisto. All right. So we have everything set up to run. I'm going to give it a try. I think that we might need to patch some of the metadata, but I'm going to click a run. And in case it doesn't work, I've got backup some file that we have run in the past that I can show the output for. So I'm going to click run. And so it's queued up in the project right now. And then, so basically what we're doing is we're waiting in line. We've kind of submitted an order. And we are asking, okay, we've got this computer project that we want to run, and we're waiting in line for a big, you know, cloud computer that's available to come available to us. And there it is. It's already ready. So now our task is being submitted up to the top, and it's beginning to run. So now I might check in with Rowan. So while this is running, Rowan, is there a way that I can edit, and I thought I saw it and I clicked for it and I couldn't find it, that I could edit metadata based on like a manifest where I can edit all the values, you know, give a unique sample ID value to each file in here? And how do I do that?
Yes, great question. So we have updated the file viewer, which is why it looks a little bit differently.
Mm-hmm.
You should be able to edit the metadata based on manifest by let's see, we're in the files tab and then the three dots there on the right, and then
Aha,
metadata
that's
with manifest. So here's the fun part. If any files are selected, that option is not available. And I'm not sure why. I think I've let them know. They're working on
okay.
it.
Got it. So that might have been why I was missing it earlier. Okay,
Exactly.
great.
It fooled me
So
as well.
okay, great. Well, now I don't feel as bad. So I'm going to select the manifest from our project. So we've already uploaded. So I'm going to select this. Now I'm going to be honest, I've never done this before. We're flying a little blind here. So, but I'm brave. So we're going to click on select and hope that it works. Hello. Not sure if anything is happening. Let me move my window. Or maybe I should try re-uploading it from my computer. Maybe that might be better.
You should be able to just select it. Did you, okay, you clicked select there.
Yes. Okay.
Okay. Ever since they
Okay.
changed, the whole file browser system, it just
Yeah.
looks differently. So
Oh,
let's,
here we go.
okay.
Here we go.
It
This looks
used to pop
familiar.
up on top, but now it's,
Yes.
below.
Pop under. So I'm going to say that yes, we want all of these metadata columns, and especially we want sample ID. So I'm going to submit that. All right. And so that gives us a progress bar. Done. And then hopefully, aha, look at that. So now we have sample IDs that have been loaded. Okay. So we're going to try to go back to our task that we ran. It is still running. And so we're going to give it a couple moments to see if it completes. And then I might try running one more time with the updated sample IDs because I think that might have been what's blocking it. And if that doesn't work, then I do have some additional, like I said, I've got some backup stuff that I could show today as well. So while that's running, let's set up another task and I'll even show you how easy it is to do that. So we can say that, oh, there it goes. So I think my problem was that I didn't have the sample IDs loaded. So it's so easy to fix your mistake that you can click this edit and rerun button. And it will pull in that same file so we don't have to go through all that steps again. And we can even call this power up too. Okay, so I'm gonna run this and we're gonna cross our fingers that this works. And if it does, then that's great. But like I said, if it doesn't, I can follow up. I believe it's a data issue in terms of how the metadata is transferred from the Kids First portal to Cubatica. So I need to follow up with some folks and look into it. So while that is, again, we're going to wait in line again, but the line is not very long because there's lots of cloud computers. So I will go, maybe we take a quick break here. Why don't we do that since we're about halfway through, and then we will pull up some output, either this output or some other output as well as part of our demo. Okay, so we are running our app. Unfortunately, I'm still having trouble with it, but I can show us some old files from another project that will show what the output looks like, and I can share what would I do if I was actually a researcher and this was happening to me. Once I share my screen again. This one. So it does give us an output message. If I was in real life and I was running this demo and trying to do this as part of my research, I might click the blue button and ask for help. Cubatica Belser also has really great office hours, which are every Thursday. You can meet with Rowan and sometimes me. I'm there every once in a while too. And they are very helpful people, can help you kind of workshop and figure out what's going on with your particular tool. So I'm going to jump over to a previous demo that I had run where I also did DESeq2. And so this is our kind of expected output. I think I did a slightly different analysis here, but you can see I'm also being able to highlight another perk of the platform is that it tracks all of your work. And so even though I ran this last summer, I still have everything right here. So just like a really good lab notebook, you want to be able to go back and be like, oh, what did I do? And what experiment and what settings did I choose? And so it's all saved here with this run. So here it's set up very similar with our different expression data files, our inputs, and our phenotype file. my settings. So this one, I think I was looking at leukemia in boys versus girls. And so we chose the covariate as gender. And then it gives us this output data that we can take a look at. So DE-seq2 is really good because it produces these output HTML files for you. Unfortunately, when I click on this, because I'm on my professional hospital managed machine here, they've determined that I'm not allowed to open that really helpful HTML file and look at the output. But that's okay, because we could back out. So normally you'd be able to click on that and you would be able to see a bunch of graphs and text and information about the different genes. But I'm going to show one final feature, which is the data studio, and how to open up this big .r data file, which has some, all of our output from this analysis. So I'm going to click over to Data Studio. So now we've made it. We're all the way over to the 5th tab now. And I'm going to create a new analysis of R Studio, and we're going to call it Power Up. And I will click the button to start the analysis. So previously, what we had been doing when we're working in the cloud is we had been kind of submitting these software requests like, hey, I want you to run this execution. And then we're in line, and then these large computers become available, and then it processes that request, and then it returns the output to us. And so it's very much like we're waiting in line, and then as soon as it, you can imagine like you're feeding a paper into the into a machine, and then it eats the paper, and then you just kind of wait for it to return something to you. So it's you kind of submit it, you wait, and then it comes back. So that's one type of cloud computing. So we're also, you can do is what's called an interactive node, or an interactive experience, where we're actually logging on, initializing what's called an instance, which is we're using this computer kind of actively. And so you can imagine we're sitting at our keyboard at our laptop in my desk, but actually the computer that's doing all of the thinking and all the processing is this very large powerful computer. So we're not using it, just briefly. We're using it kind of continuously, and allows me to be able to kind of insert multiple smaller commands instead of just having to, copy everything all at once and have the machines run it all on its own. So it is initializing that computer. And so what it's going to do is it's going to load up this environment called R Studio, which many of you are familiar with. If you're not familiar with R Studio, so R is a kind of a data analysis language tool that allows you to be able to graph, do statistical analysis, and be able to kind of tear apart data and produce findings and results that you might learn about. So R Studio you can load on your your local machine, your laptop, and you can carry out analyses. Depending on the scale of it, that may be sufficient for what you need to do. And so if you only have very relatively small files, then maybe you can run it on your laptop and you can carry out these types of analyses. But if you're looking to do a much larger kind of scale type of thing, where you're looking at a very big analysis with lots of data set, you can use the Data Studio on Gravatica, which will have the same, once it loads up here, almost there, same kind of R Studio window, ta-da, just like you would have on your laptop. But now, instead of it running on my laptop, the computer, the chip in my computer is not doing the processing. Instead, the computer in the cloud is doing the processing. So we're interested in in those files that exist in our project. And so if we kind of switch over to a terminal view, we can do an ls and just kind of see we're in this empty file. But I can go, because I know a little bit about the nature of our, how the structure is here. So it's actually in up a directory in a thing called project files. And then it returns all of this output. And so we are particularly interested in this, let's see here. So this was for a demo for ASG. So we're interested in this ASG demo.r data file. So this is where all of our graphs and stuff have been kind of bundled together as output from DE-seq, and we want to open that up here in R. So this is not an R course, so I'm not going to go into too much detail about how to use it, but I just want to demonstrate the types of stuff you can do. If you're interested in learning more about R, we can share some places where you can go to kind of learn some basics about the tools. So if I go to load, and I'm going to load that file, and I might have done it wrong. I might not need those quotes. Let's see here. What am I doing wrong? I think I might have gotten the file name wrong. That's what I got wrong. Copy. All right, let's try that again. Because now I have the wrong directory. If I do // project files / there we go. Third time's a charm. So it's going to load up that our data file. And so basically what it's going to do, it's going to unpack all of this output that we generated from our DE-seq2 workflow. And as it refreshes there's our big kind of collection of information. And so let's see here. So we've got a couple different files that we have available. So I believe the file RES, so this is this big kind of output file where it describes is so large, it's not going to, it's 54,000 rows, which corresponds to 1 row for each gene, and then it gives us information about the expression of those genes. And I can share, information about how you might want to do an analysis, but just to show a quick, kind of plot of this this type, and we can scroll down. And we've got to load our package here. So because it doesn't recognize that command. So if we load in the DE-seq2 package, and then we do our plot, ta-da, so there's our plot. And so what it shows, what we're looking at here is we're looking at along the, and it refreshes and makes it look pretty, along the x-axis, this is the mean of normalized counts. This is how highly expressed the particular gene is. And then on the y-axis, this is the relative expression levels in our 2 different conditions. So in my example, I was showing this would be ependymoma, high in ependymoma versus high in medulloblastoma. I believe in this one, like I said, this is high in males versus high in females. But then this would allow you to then, this is kind of the first overview. So you can see that there are a handful of genes that are very highly expressed, more than my limit here. In fact, I could even expand the limit if I really wanted to. And so those might be interesting candidates that I might want to explore. And so we could then pull those out of that table, that RES table, what are these really high numbers, and kind of dive in a little bit further. So I'm not going to kind of explore that. I can share some resources about R, about where you would want to go next. But do want to highlight, the types of things that you can do on the platform and where you can go. All right. So now I do want to show in my last bit here that I want to show is I want to show a little bit about how do we connect GTEx data with this expression data. And so we have a couple different ways that you can do that. And so if I go up to the top here and we navigate to the GTEx portal, and it will load up. And so we have information about our particular files. Yes, Tim, yes. So I think maybe you weren't here at the very beginning, but I shared the directions to the tutorial that I was following today, which does go into more detail about, kind of further downstream about R. And so the directions I've been following will be available, and then we are going to share the recordings too, so that will be available for you to follow along. So here is our the GTEx portal, which has GTEx data in it. And so they also have RNA-seq data. And so it could be valuable. So for example, you can imagine that, well, maybe I want to compare. So in this, our demonstration that we did today, we looked at medulloblastoma, which is a tumor of the cerebellum. and we compared it to ependymoma, which is a tumor of the epindal cells. And so it might be good to, well, maybe wouldn't it be better if we could compare medulloblastoma to healthy cerebellum tissue? And so that might give us better insight, not necessarily to the difference between those two types of tumors, but maybe what specifically about medulloblastoma is upregulated or upexpressed might make that particular type of tumor possible. So we can do that. You could do that a couple different ways. So here are our data for the GTEx analysis. So it has a couple different file formats. But if we look at gene TPMs by tissue, this is kind of an adjusted format. And then each of these are these different tissues that I think I introduced all the way back on Monday. We can go back to our tissue sampling site. And so one of the tissues that's available is cerebellum. And so we can look for just expression values that correspond to the cerebellum tissue. So I'll show a couple ways that we can integrate this. I'll show 2 ways we can integrate this. First way you can do this is that we're going to go to our find our cerebellum file. Let's see here. If I go back, brain cerebellum. So this is our gene expression value for brain cerebellum. And so what I could do is a couple ways. So I could download this to my computer and then upload it into Cavatica using that method that I showed you. I'm actually going to do a wget and show that we can actually copy this file right into our interactive node. So I'm going to copy the link, and we're going to go here, and we're going to go back to Cavatica. And so I'm going to go back to my terminal, and I'm going to, clear it out. And we'll do an ls, and so there's nothing in our directory, and I'm going to do a wget, and I'm going to paste that URL. So what wget is basically, it's like download this file and save a copy of it to this location. So now if I do ls, then I can see I've got this gene expression in the cerebellum. So right now it's zipped up. So if I do, head on this file, it's just going to return, I'm even afraid to do this, it's going to return the gobbledygook because it's a compressed file. And so instead, what we're going to do is we're going to unzip it. Let me clear again. There we go. We're going to unzip it using g unzip, and then we're going to run that file. And so now if I do ls-l, so here's this file, gene TPM, and now it's unzipped. It's just a GCT. So again, I can do, if I just want to see what this looks like, I can type head, and so it returns. So what we're looking at is a really, really big table. The tables are not really great to look at on the terminal, but R is pretty good at pulling up tables. So we can actually go back to our console here, and we can load that file that we just pulled up, if I remember the name of it. Or I can import it. Maybe we can do an import. Let's do import from our text file, import, open, and then here's our file format, approve. And it's taking quite a bit because there's 56,000 lines, but there it goes. And so then here's my GTEx data. So this is in that, it's in that particular, it's .gct format. So the first 2 rows, the first row is kind of information about the number of samples and the number of participants. And then the second row is a descriptor column. But then in our V2 column here is each individual gene. And then V3 is the kind of the shorthand gene name. And then these kind of following columns after that are expression values. And so if I go back to, let's do this, there we go. Each of these columns corresponds to a different particular sample. And so we know each of these is cerebellum, and then we could link it back based on our expression values there. if we wanted to kind of compare. We could also split this file into individual files, and then you could feed those into DE-seq as well. So that would be one option. So what we just did was we copied this file from, we downloaded it, and so we saved a copy of it in our cloud workspace. And so that's one method to kind of get this data. So I'm actually going to stop my analysis here. So I'm going to show the other way that we can load GTEx data in Cavatica. So we're going to stop like that. Why does GCT still exist? Great question, Tim. That is a question that I was wondering as I was putting this demo together myself. I would love it if we had individual files for every single sample, but that's what we've got. Great question. Okay, so that's kind of my analysis. So I've saved that. So I've basically, I said, thanks for letting me borrow your computer. I'm done using it now. Flip the off switch. And so it bills me. So that cost me 11 cents to run that. To use the computer for that period of time. And so obviously it's kind of like the lights in your house. Like if you leave the room, you want to turn the lights off. And so otherwise you're going to run up your power bill. And so similar to the submitting the tasks, which also, you're billed based on how long you're using the computer. When you're not using it, you want to turn the computer off. And like I said, CFDE does support cloud credits to be able to do that. So that way you don't have to pay the 11 cents. NIH will pay the 11 cents, which is even better. Okay, so that's one method. So now I'm going to show another method of pulling GTEx data into Cavatica for a combined analysis. So for this, I'm going to go to, so if we explore the GTEx portal a little further, and we go into home or downloads, tells us, note, GTEx protected access data files are also housed in Anvil, not the sequence read archive. So GTEx has a lot of different types of data. It has kind of two main categories, similar to Kids First, where there's open access data that anyone can use without having to fill out a form or permissions. And then there's also protected access, which requires a dbGaP application, explaining what you're looking for. So this is like the alignment files that we were talking about. There's also a whole genome information from GTEx as well. So you can apply for access to that. So we're just going to try to look for some RNA kind of expression data as well. And so it's housed on Anvil. If we click this link, it'll take us to the Terra Anvil. So we actually want to go to our Gen 3 Anvil, which is available at this URL right here. The Anvil is another kind of DCC type of organization where they help to do this large data scale data science analytics space. And so we want to be able to copy files from here and bring them into Cavatica where we can combine them together. So I'm going to log in. So again, similar to like Kids First, you can log in with an ERA Commons, or you can log in with your Kids First ID. And we're going to sign in. And it makes available to us a couple different files, downloadables, things like that. So they have a couple different data sets. So I'm just going to highlight two. So one thing that they have in here is they have the 1,000 genomes data. So if you're familiar with that, 1,000 genomes is this really cool project where they sequenced the whole genome of 1,000 individuals from around the world to really try to reflect, the diversity of human biology, which is awesome. And so that data is open access and available. Is kind of the, I don't know if it was officially the inspiration, but I think it was the inspiration for the All of Us project, which is now ongoing, based out of the United States, which has a similar mission of, sequencing a huge cohort of individuals to try to, allow for very large scale analyses. So the 1,000 genomes data is in here as well. We could copy that to Cavatica, but we're not going to do that today. Today we're going to work on GTEx, which has information from 981 particular subjects. So we might go to our Files tab. And again, we're going to narrow it down to our GTEx data. And so we have 122,000 files, which is a whole lot. So we can kind of narrow down a little bit further, really similar to Kids First, where we, kind of looking for just the specific files that we're looking for. And so they have a couple different file output types here, similar to Kids First. So we could select, specific expression files, or if we wanted to start at the beginning, if we had our own tool, maybe we want to start with the alignments and we want to run them, ourselves. So that would be one option. You could move the alignments, or you could look at, expression data as well as some of these output files and move them too. So all these are very large cohorts. So just for the sake of our demo today, I'm just going to pull out some of these analysis files. And so these are kind of like metadata files to help you explain, what is GTEx, what are all these individuals, what are what are the age of these particular participants, things like that. It's also a nice, tidy, neat 17 files, which will transfer hopefully very quickly here in the closing couple seconds of our demo. So I've got these 17 files selected, and so I have the option to export to 7 bridges. So I'm going to click this button, and there's our Cavatica, there's our tool. So I'm going to click export to Cavatica. So it's going to take a couple moments to think about that. It says that we're in progress. And so while we're exporting here, I can highlight. So the what I showed earlier with the GTEx using the wget, what I did was I copied that file and I pasted it into my Cavatica project. And so what we did, so I said what we've been trying to do is we don't want to do that, right? We don't want to copy and paste. We want to have pointers, so that way we're not, proliferating more and more copies of these files, which results in more things being stored, more versions of the same thing, and that can lead to more cloud costs as well. So a better way to do it is to use this interoperable method, which I'm showing here, where I'm kind of connecting, I'm importing this data. It says you're about to import data from the Gen 3 Anvil as DIRF files with their metadata. And I say, yep, that's great. We're going to put them in our Power Up demo project. And we can take these as we'll just have this as GTEx. And we're going to say that I understand that I'm going to use the data responsibly. And we're going to click that button. And it's going to import start successfully. And because we chose such a small number of files for our demonstration, if I go over to files, I can even click and learn on the little bell and learn about the status. So we're still preparing to import to our Power Up demo for Gen 3 Anvil. So we'll give it another couple minutes for the computers. Basically what we're doing is similar to, these files that we pulled as already done. So what are these files from the Kids First? We didn't copy these files. We are just, accessing them in another location. And so if I hit refresh, and now we have here are those GTEx files. So we've got our GTEx data, which is loaded up. And so then we can review that as well. So this could give us information. So for example, the subject phenotypes, which I guess I could have, if I had done my interactive node, if I still had that up, I could pull up the subject phenotypes file. What this does is it tells you more information about, I don't think it'll let me do the raw view of a text file, it won't. It gives you the information about the, who are these people, that code of those, the table of GTEx individuals. So maybe, like for example, Kids First is just pediatric cases, whereas GTEx includes adults of many different ages. So maybe I just want the GTEx of individuals, the GTEx data of individuals who are under 30, because that might be closest to the, the pediatric participants. So you can use the tables here to kind of help filter out that analysis. All right. So I showed a couple different things today. So let me stop sharing and let me pull up my screen again and bring it in for a conclusion. So hopefully today, what you can take home with you is, if I do the right button right there. We identified some RNA sequencing expression files on the Kids First portal. We showed how we pushed them to Cavatica, and we ran our DESeq2 app, and then we were able to view output in RStudio. And then I showed a couple different ways that you can combine GTEx data and bring it into Cavatica as well. So that way you could kind of scale your analysis up and combine the two different DCCs together. All right, and so then we're going to close as we began. So we're going to look back at our calendar. So today was, kind of using the existing tools, kind of, getting our feet wet and exploring what types of things are out there. And so tomorrow we're going to, scale ever more boldly into even bigger tasks. So we're going to be building our own pipeline. So rather than just, using the existing DESeq2, Sangeeta is going to show how she's going to build her own tool that does her own analysis that she wants to use. So, not just using the existing apps, but building her own. All right. So with that, I think I'm going to close here for today. So thanks so much for joining. We will follow up. I can try to follow up this afternoon after our course with some of the materials that we use, just as kind of a follow-up, the links from the chat that I shared. And yes, we will try to make the recordings available as well afterward. So with that, I'm going to close here. I'm going to say thanks so much for joining today, everybody. I hope to see you again back here tomorrow at 10 a.m. Eastern time, where we're going to learn how to build our own workflow. So it'll be really exciting to do the next step. And thanks again to Rowan for coming in and helping out today. I truly appreciate the on-the-fly help and support.
Happy to be here.
Awesome. All right. Well, thanks, everybody. Have a good day. We'll see you tomorrow.